Hello students. Today we start with next point, that is 6.9 Human Genome Project. Under this point, the subpoints are 6.9.1 Salient Features of Human Genome, 6.9.2 Application and Future Challenges. So these two subpoints we have to study under Human Genome Project. These points are from Chapter 6, Molecular Basis of Inheritance, which comes under Unit 7, Genetics and Evolution. Human Genome Project. This project was launched in the year 1990 to sequence human genome. Genome, the whole hereditary information of an organism that is encoded in the DNA. The project is called Mega Project due to the following reasons. First, human genome is mainly consisting of 3 into 10 raised to 9 base pairs approximately. Cost of a single base pair sequence, that is per base pair $3, then estimated cost for the project will be 9 billion US dollars. Second reason, obtain sequences if typed in a book to store with 1000 letters per page, 1000 pages per book, then 3300 books are required to store the information of DNA sequence from a single human cell. And third is enormous data expected to generate require high-speed computational devices for data storage and retrieval and analysis. Because of these reasons, the project is called Mega Project. Here you have to remember HGP, that is Human Genome Project, was launched in the year 1990 and completed in the year 2003. 13 years required. And the major partners for this project were US Department of Energy and National Institute of Health. The total cost of the project would be approximately 9 billion US dollars. And to manage enormous biological data, a new branch of biology has been developed that is called as bioinformatics. So all these are the points you have to remember why the project is called Mega Project. Next point is important goals of HGP. First, identify all the approximately 20,000 to 25,000 genes in human DNA. Second, to determine the sequence of 3 billion chemical base pairs that make up human DNA. Third, store this information in databases. Fourth one is improve tools for data analysis. Fifth one, transfer related technologies to other sectors such as industries. And the last is to address the ethical, legal, and social issues that may arise from the project. So these are important goals of HGP. Our next point is salient features of human genome. The human genome contains 3164.7 million nucleotide bases. Second, the average gene consists of 3000 bases, but sizes vary greatly, with the largest known human gene being dystrophin at 2.4 million bases. The third, the total number of genes is estimated at 30,000, much lower than the previous estimation 80,000 to 140,000 genes. Almost all 99.9% nucleotide bases are exactly same in all people. So these are the important features of human genome. Next is the functions are unknown for over 50% of the discovered genes. Less than 2% of the genome codes for protein. Repeated sequence make up a very large portion of the human genome. Next is repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes 100 to 1000 times. Next feature is chromosome 1 has most genes, that is 2968, and Y chromosome has the fewest genes, that is 231. So one mark question may come, the number of genes present in chromosome 1 and chromosome Y. So you have to remember the numbers, that is chromosome 1 has most of the genes 2968, that is, and chromosome Y has few genes 231, maximum and minimum. Scientists have identified about 1.4 million locations where single base DNA differences, that is, the single nucleotide polymorphism, and it occurs in the human. So scientists have identified the location where the single base DNA differences occur in the human. So these are the important characteristics of human genome. Our next point is applications and future challenges. So here the knowledge of human genome sequence and research will lead us to understand the biological system in coming decades. HG sequence will enable a complete new approach to biological research. With whole genome sequence, a new high throughput technologies, we can approach questions systematically and on a much broader scale. So these are the applications. So particularly due to this knowledge of this human genome, it helps to complete understanding of the human biology and the genetics. Then many genes associated with human disease were identified, and it helps in understanding diseases, development, and new method of diagnosis and treatment. So these are all applications of the human genome. And again, you will find that it will possible to understand how enormous number of genes and proteins work together in interconnected networks in the chemistry of the life. And also HG sequences is enormous task which will require the expertise and creativity of tens of thousands of scientists from varied disciplines in both public and private sectors worldwide. So this point will give you an idea regarding the applications of human genome project. So in this lecture, we have studied in brief what is mean by the HGP, what are the goals of the HGP, what are the important features of human genome, and how important the knowledge regarding the understanding the different diseases and to apply the new treatment to cure the diseases. Thank you.